Thank you all for coming today and joining us for our news conference on endocrine-disrupting chemicals. I hope that you're enjoying your time at ENDO so far. So I am Joanna Spencer-Segal, and I'm a member of the Endocrine Society's Advocacy and Public Outreach Core Committee. So a couple things about me. I received my PhD from the Rockefeller University working on how estrogens affect memory formation in the brain. I also have an MD, and I am currently one of the Society's in-training members, just a few days ago, having completed my residency in internal medicine at the University of Michigan, and I will start a fellowship in endocrinology there in July. So that's me. And so today, as you know, the topic of our conference is endocrine-disrupting chemicals, and I just wanted to give you an idea of what we're talking about there with EDCs. The Endocrine Society is proud to be one of the leaders in just getting information out to the scientific community and to the public about endocrine-disrupting chemicals. And we have a scientific statement on this topic that if you haven't seen, our staff can help you find. But in that statement, we define what an endocrine-disrupting chemical is. And so I'm going to read you that definition. It's a compound, either natural or synthetic, which through environmental or inappropriate developmental exposures alters the hormonal and homeostatic systems that enable the organism to communicate with and respond to its environment. And so what this means, natural or synthetic compound, meaning compounds that are found in plastics or toiletries or pesticides, or even in foods like the phytoestrogens found in soy. All of these compounds can be considered endocrine-disrupting chemicals, and we can get exposed to them through the food chain or through leaching from plastics and or through using products. They get into our bodies and they bind to either hormone receptors or neurotransmitter receptors, or can also alter the synthesis of hormones in the body, and in this way disrupt many different systems in the body. And some of the systems that have been implicated in endocrine disruption include reproduction for both males and females, also the development of hormone-sensitive cancers such as breast cancer, prostate cancer, disruption of the neuroendocrine system, of thyroid function, and am I missing one? I'm sure I am. So as you can see, their effects are broad-ranging. So that's what we're going to talk about today, and I just want to introduce you to today's speakers. We have Dr. Gail Prince. Dr. Prince is the Michael Reese Professor of Urology in the Departments of Urology and Physiology and Biophysics at the University of Illinois at Chicago. And Dr. Prince was also one of the authors of the Endocrine Society's scientific statement on EDCs. Dr. Patrick Fenichel, he is the professor and head of the Department of Endocrinology at Centre Hospitalier Universitaire de Nice in Nice, France. Ms. Rebecca Kennedy. Ms. Kennedy is a graduate student in the Department of Public Health at the University of Tennessee, Knoxville. And finally, Dr. Almudena Vega-Lopez is a research investigator in the University of Michigan's Environmental Health Science Core Center's Department of Pediatrics. So these are our speakers. We're thrilled to have them with us today. And this is how the conference will run. I'll have each of the speakers come up and present their work. And then after all the presentations have finished, we'll do questions from the audience. Please note that we are broadcasting this online live, and there are many journalists who are with us online. What that means is that everybody needs to make their remarks into a microphone so that those who are joining us online can also hear. And journalists who are joining online will also be able to answer questions, ask questions through typing. Okay? So with that, I would like to invite our first presenter, Dr. Prince.
Okay, thank you for coming this morning to hear about our work on endocrine-disrupting chemicals in our research on that topic within the Endocrine Society. And I'm going to show you quickly and highlight the work that we'll be presenting today at the endocrine meetings on early life or developmental exposure to low doses of bisphenol A and how it can increase the carcinogenic risk or susceptibility of human prostate epithelium. And this will be presented by Wen-Yang Hu at this morning's session at 11:15. So this work sits within an overreaching paradigm of the fetal basis of adult disease. And it's been well established now that many adult diseases now have their origin during the developmental period, where susceptibility to things like diet and stress and environmental chemicals can have their greatest effects. And this has been established for heart disease, diabetes, and others. And within this context, the research from my laboratory for a few decades now has asked the question whether the risk of prostate cancer can be determined in the womb. And so what we've done for over 20 years is we've looked at one of the hormones that is at high levels in the womb, and that is the mother's estrogen levels, or estradiol, as well as pharmaceutical exposures. And we found that indeed abnormal exposures to estrogen during development can in part explain the very high rates of prostate cancer that are seen in aging men. So for the past 10 plus years, we've been also looking at endocrine disrupting chemicals and what they might do during this similar period of time. And the chemical that we'll be talking about, and we've done a lot of work on, is bisphenol A, which mimics estrogen. It's a high volume chemical at very high levels in a number of personal products and well, we don't even know where they all are, but the ones that come out most frequently are polycarbonate plastics that have BPA in them, epoxy resins of can linings. They're within carbonless paper receipts, and most humans are exposed to BPA on a chronic basis. Even though it's rapidly cleared from the body, most humans measure positive, so we know that it's rather ubiquitous in the environment. So to see whether this might influence prostate cancer risk, our initial model was to look in the Sprague-Dawley rat, and we gave a very brief exposure to BPA on days 1, 3, and 5 of life when the prostate gland develops. And what we found was that it did not affect prostate cancer or drive prostate cancer directly. But when we elevated the estrogen levels, and this occurs in all aging men, their estrogen levels start to rise. And when we do that, plus give the BPA early in life, what we find is that the early life exposure to BPA over in right increases the susceptibility or risk for developing prostate cancer. So if we look at BPA as compared to C there, it significantly increased prostate intraepithelial neoplasia, the precursor lesion for prostate cancer. So one of the major remaining questions is this relevant for the human prostate gland? gland. And so to address that, what we've been doing in our laboratory is we've been taking prostate epithelial cells directly from patient specimens in the operating room or from organ donors at the time that their organs are harvested due to an untimely death. And in the latter instance, these are disease-free prostate specimens. And using new technologies, we've been able to culture out the stem and progenitor cell populations from these cells and get large numbers of them for our study. And so these stem cells are pliable and can be used to grow and totally regrow new prostate glands. And so what we do, and we have done in this unique model, is we take these prostate stem cells and we graft them under the kidney capsule of adult male mice. And within 1 month period of time, they form little prostate structures under the kidney capsule, and by careful staining for prostate-specific antigen, PSA, the human prostate marker, we can see that these are human prostate-like structures that are growing in these mice. And so if we give them elevated estrogen, then we can see that they develop prostate cancer, but at a very low rate. So what we've done in our presentation that we'll be showing you today is when these prostate structures are being formed in the kidney, in these grafts, we give a low-dose exposure to bisphenol A for the first 2 weeks. We do it orally to these mice. We've measured the free circulating unconjugated BPA levels. They're very low, and they're similar or the same as levels that are found in the human serum. That is about 0.16 nanograms per mil or 0.45. So these are very low levels of BPA. We did another group where we gave BPA as we're culturing the stem cells in addition to the BPA in vivo. And we do that for only 2 weeks, discontinue it, and then give the estrogen in adulthood. And this is the final data slide. What we've been able to find is in nearly 120 grafts that we've looked at, if they received only control vehicle, they get a prostate cancer incidence of 12%. If they saw BPA during the development in vivo of these humanized prostate structures, it went up significantly to 33%. And if they had BPA during stem cell growth as well as during prostate development, it went up to 45% prostate cancer. And so we conclude that our new findings provide the first direct in vivo evidence that early life exposure to BPA at levels found every day in humans increases cancer susceptibility in the human prostate epithelium. And this work was funded by the National Institute of Environmental Health and Sciences. Thank you.
Thank you very much, Dr. Prince. Now I would like to invite Dr. Fenichel to the podium.
Thank you. Good morning. As Professor Prince told you, there are now good evidence to think that fetal exposure to chemical with estrogenic or anti-androgenic effects can help to develop adult disease like cancer. But it's quite more difficult to demonstrate that in humans. But we have no reason to think that it will not be the same. But clinical studies, as I will show you, is quite more difficult to perform than experiments in rodents. So we have been interested in our team in cryptorchidism. What is cryptorchidism? Cryptorchidism is undescended testis at birth, and it is the most frequent male congenital malformation. It occurs between 2 and 6% of full-term newborns. It can reverse spontaneously in 50% of the cases before 1 year, but in all cases, reversible or not reversible, you have an increased risk in adulthood of hypofertility and testicular germ cell cancer. For this reason, we are interested in this pathology, which in most cases remain idiopathic, isolated, non-syndromic. Just to explain how will occur the physiological testicular descent. You have 2 ligaments, 2 phases during pregnancy, and 2 Leydig hormones who will regulate this descent. The first phase is during the second trimester of gestation. It's called the transabdominal descent. The second phase is the inguinoscrotal phase, will occur at the end of the pregnancy. And this descent will be controlled by these 2 hormones which express receptor on the 2 ligaments. And there are 2 important hormones, insulin-like peptide 3 and testosterone. They are both involved in, for insulin-like peptide 3 in the 2 phases and for testosterone in the last phase. There are mutations which have been described in humans of these 2 hormones and the receptor associated to cryptorchidism. But these mutations are rare. For this reason, it has been suggested that these chemicals with estrogenic or anti-androgenic effect could impair the secretion or the action of these 2 hormones and induce cryptorchidism. This hypothesis, called the environmental hypothesis, is supported by wildlife observation. You know the panthers of Florida, with 70% of cryptorchidism in an environment of using pesticide and PCB. You have experimental models. It's very easy in rodents to induce cryptorchidism with phthalate or distilben or other estrogenic or anti-androgenic chemicals. But in human, the evidence are less important. We have some indirect evidence, which is the increasing incidence of cryptorchidism, geographical difference between areas, and you have some indirect relation between concentration of these chemicals and cryptorchidism. We have conducted a prospective case control study on isolated idiopathic cryptorchidism in the area of Nice, south of France, during 3 years, 2002 to 2005, and we have systematically screened all newborn in the University Hospital of Nice, 6,000 newborn male, with a standardized neonatal examination. For each case, we have chosen 2 control matched boys, and we have collected cord blood and maternal milk, and we have measured INSL3 testosterone, the 2 hormones, free bisphenol A, we have been talking about, DDE, PCB, and phthalate. And I show you just 3 slides with the result, which have been presented yesterday in a session of the Endocrine Society. First of all, we can see that the levels, the cord blood levels of INSL3 are decreased when compared to the control. And they are mostly decreased in transient cryptorchidism. So, for the first time, we demonstrate that a major actor of the testicular descent is decreased in cord blood in cryptorchidism. Secondly, the second result is we have looked at the relation between level of INSL3 and bisphenol A. And what we show is a very strong correlation between bisphenol A and INSL3. The highest the BPA level was, the lowest the level of INSL3 was. So you have an indirect negative relation between the two, and you can suggest that bisphenol A, which has an estrogenic effect, may repress the expression of INSL3 and induce cryptorchidism, or help to induce with other factors cryptorchidism. So, to conclude, we have not demonstrated that bisphenol A is the only factor which induces cryptorchidism, but this clinical study suggests that fetal exposure to bisphenol A will help to decrease INSL3, a major actor of the physiological testicular descent, and will represent a cofactor which with other factors, other endocrine disruptors, or other elements such genetic susceptibility or anatomical factor will induce cryptorchidism, idiopathic cryptorchidism. Thank you.
Thank you, Dr. Fenichel. I now invite Ms. Kennedy to the podium. Okay, hello everyone. I'm going to switch things up here and talk about triclocarban today. So I wanted to talk about how triclocarban exposure during lactation alters survival rate in the female rat neonate. And I have no conflicts to disclose. So I wanted to begin with a background to triclocarban. Triclocarban is an antimicrobial compound found in personal care products, and it's primarily found in bar soaps. It's a high production volume chemical produced at 500,000 to 1 million pounds per year. It's widespread and persistent in the environment. Once it's washed down the drain, it's discharged into the environment through incomplete wastewater treatment. It has a long half-life of 87 to 231 days in sludge-treated soil. And it's also detected in aquatic and terrestrial species, and it's been found in the earthworm as well as fish, and then sometimes plants meant for human consumption like the zucchini or the pumpkin. Now, primarily, humans are exposed to TCC through direct dermal contact, where it's then washed down, where it is then absorbed through the skin. And this figure here evidenced the skin absorption with peak concentrations of blood levels at 2 to 3 hours after a 15-minute whole body shower with TCC-containing soap. Now we know that research has evidenced that TCC has the potential to artificially amplify male sex accessory organ weight in the adult male rat. However, at this point, the impact of early exposure is currently elusive. Now, Dr. Barker has shown that insults during early life can contribute to poor health outcomes later in life. And thus we hypothesize that TCC exposure during pregnancy and or the nursing period will alter future development. So initially we created a study design where we looked at TCC exposure during pregnancy as well as during the nursing period. And so 2 groups of pregnant rats were divided, where one was provided just chow food and the other was provided chow food supplemented with 0.5% TCC. And then they were followed through until the weaning period throughout the nursing period. And we found that there was reduced survival as among the neonates born to and nursed by the TCC supplemented dams, the survival was reduced to postnatal day 8. So we wanted to know what was the critical period of exposure.
Was it during pregnancy? Was it during the nursing period? Or was it a combination of both? And so we had the objective to determine the critical period of TCC exposure related to survival in the female rat pup. So to determine this objective, we created a crossover design. And the initial design was similar. However, at birth, we implemented a crossover. And so each female rat nursed 2 of her own pups and then 2 pups from each of the 2 other treatment groups. And so these pups had exposure during the pregnancy as well as during the nursing period. These pups had exposure only during the nursing period. And these pups had exposure only during the pregnancy period. And so in this manner, we could see exposure during different stages of development. So over the course of this study, we looked at TCC levels in the maternal blood as well as the amniotic fluid. Ultimately, we looked at survival. We looked at body weight gain or weight loss over the course of the study. And then we also looked at milk production. So we found a dose-dependent increase in the concentration of TCC in the maternal blood as well as the amniotic fluid based on the dose provided to the maternal rat. And I want to emphasize that the maternal concentration that we found in the maternal group supplemented with the highest concentration of TCC is similar to concentrations found in a chronic user of TCC after a whole body shower with TCC containing soap. So we found, regardless of the group that the animals were born to, there was reduced survival when pups were nursed by TCC supplemented dams. So in the first figure here, you can see that only 4 out of 30 pups survived until the weaning day when they were nursed by 0.2% supplemented rats. Now the situation was even more profound among pups nursed by 0.5% rats, as no pups in this group survived beyond day 5. However, when the pups were raised by control rats, again, regardless of the group that they were born to, their survival was maintained across the course of the study. And so collectively, this indicates that when that lactation, or the nursing period, is the critical period for survival related to TCC exposure, because it didn't matter what group the pups were born to, as long as they were nursed by the TCC-treated rats, their survival was reduced, and as long as they were nursed by the control rats, their survival was maintained. So trends in body weight mimic that of survival over the study. Now, as early as day 3 of life, we found a reduction in the body weight of neonates nursed by TCC supplemented dams. Now, interestingly, while pups nursed by control rats were able to survive across the course of the study, if they were born to 0.5% supplemented rats, their body weight was reduced compared to the 2 other groups as early as postnatal day 3, and this was a trend that continued until weaning. Now, so we wanted to know, did TCC affect milk production, and was that the cause of the reduced survival that we found? However, we found evidence of milk bans in the pups provided to TCC-exposed rats during the nursing period, and you can see that here. So this indicates that the rats were able to produce the milk as well as the pups were able to obtain the milk. So then we wanted to know, was it possible that TCC affected the health of the pups, thereby reducing the suckle response and indirectly affecting milk production? However, even when healthy, stronger control pups were added into a TCC-treated cage, neonates born to and nursed by the TCC-treated dams still couldn't survive, even with the suckle response maintained. This indicates that some other mechanism was coming into play, which we are currently working on what that mechanism is. So I just wanted to conclude with showing that TCC exposure during early development decreases survival rate. And we showed this initially where pups were exposed during pregnancy as well as during the nursing period. And then we feel confident saying that the critical period for survival is dependent on exposure during the nursing period because it didn't matter what group the pups were born to. If they were nursed by TCC supplemented dams, their survival was reduced, and if they were nursed by control dams, their survival was maintained. So I just want to acknowledge everyone that helped me on this project, and then thank the National Institute of Environmental Health Sciences for funding the research. Thank you.
Thank you very much, Ms. Kennedy. For our final presentation, Dr. Vega-Lopez.
Good morning, everyone. So I would like to thank the Endocrine Society for selecting our work as newsworthy and for you guys be here. So the work I'm going to present today was done at the pediatric department at the University of Michigan, and I'm going to be talking about the interaction of prenatal bisphenol A treatment and postnatal overfeeding on insulin sensitivity and inflammatory pathways in visceral as well as subcutaneous adipose tissue depots with nothing to disclose. So the previous presenters have already pointed out that any impact on very early on that any organism is exposed to can lead to adult disease or even make you more susceptible to develop adult diseases. And we've already talked about the fact that humans are exposed to all kind of different endocrine disruptors, and that those are just mimicking the effects of endogenous hormones. And one of such endocrine disruptors is bisphenol A, which is a component of polycarbonate plastics and epoxy resins, and it's present in many different consumer products. And the importance of bisphenol A is an estrogen mimic. So it can mimic what endogenous estrogen does. So bisphenol A and estrogen are two keys to the same lock, which is the estrogen receptor. And this is quite important since estrogen receptor is present in many different tissues in the body. And that's why it's been found that prenatal exposure to BPA can lead to a variety of disease outcomes. And that I just want to briefly mention on three of these. It's been shown to lead to impair insulin homeostasis, which is the way that the body regulates glucose and insulin. It's been shown to lead to immune dysfunction, or put in other words, the way the body fights against infection, and it can also affect the way adipose tissue develops. So any environmental insult, in our case BPA, can affect fat function through two different mechanisms. One would be I can increase the number of immune cells that will infiltrate the fat, and these cells are called macrophages, or it can also induce a reduction in a key hormone, which is adiponectin, and adiponectin is essential to insulin regulation. So you want your adiponectin levels to be high. And this, the infiltration of macrophages as well as reduction in adiponectin can lead to fat inflammation, which can lead then into insulin resistance, which is a pre-state of type 2 diabetes and a component of metabolic syndrome. So what there's another factor. Overfeeding can impair fat function. The more a person eats, the more fat it's going to deposit. So what we thought was that we wanted to test whether prenatal exposure to BPA will lead to insulin resistance, and if that would be through the two mechanisms that we talked about, increased macrophage infiltration and reduction in adiponectin. And for that, we used our an animal model, a large animal model. Most of the studies that are out there are in rodent models. So we need to use either non-human primates or large animal models that can ultimately translate to humans. So we used the sheep, and our study consisted in the administration of bisphenol A during mid-gestation, and the dose we achieved was similar to, the internal dose achieved was similar to human levels, and those, the offspring of the mothers treated with BPA were put on a dietary manipulation, either on maintenance or they were overfed, and we did a glucose tolerance test to check for their insulin homeostasis when they were young adults, and then we collected adipose tissue samples from two depots, from the subcutaneous, which is under the skin, and from the visceral, which is the belly fat. And these are just briefly the results that we, from the glucose tolerance test. We didn't see a specific bisphenol A effect in the baseline or the morning insulin, but we found an increase in morning insulin levels in both overfed groups. And we didn't see a difference in the response to the glucose challenge in any of the 4 groups studied. And then we looked at the both subcutaneous and visceral fat depots. We did gene expression studies, and we found a specific. You can see to the left of the slide that the subcutaneous depot, we found an increase in CD68, which is an inflammatory. It's an inflammatory marker. It's a marker for macrophage infiltration. We found an increase in maintenance fed animals treated with BPA, as well as a reduction in adiponectin, but only restricted for the, to the BPA group, indicating an interaction between BPA and overfeeding. And then in the visceral fat depot, we did not find any BPA-specific effect, but we found an overfeeding effect, finding an increase in CD68, the inflammatory marker, as well as reduction in adiponectin, which is what we think is driving the hyperinsulinemia that we found in these 2 overfed groups. So we can say that prenatal bisphenol A and diet may interact to modulate the inflammatory mechanisms that we see within these 2 fat depots, but in a different manner, and we're trying to dissect that out at the moment. And with this, I would like to thank all the participants in the study and our NIH funding. Thank you.
Thank you very much, Dr. Vega-Lopez. I think it's really a fantastic variety of science that you've seen today. A couple things I just want to point out to you in the science of EDCs. As Dr. Finichel pointed out, one of the challenges in this field is really to do science in the lab that's relevant to human exposure, and it's very difficult, it's impossible to do controlled human trials on exposure to these chemicals. And so with that, you know, I think you've seen some really creative approaches today using whether that's very well-designed human studies or human cells in an animal model, or going to larger animal models to very, very creative approaches dedicated to really doing science that's relevant to human health. So I think that's great. Also, just to point out that all the studies that you've seen today are really looking at the effects of EDCs during development on later development of disease, and that's really striking, because what that means is for humans that are born that already have had exposures to these compounds, that perhaps some of the risk is already there, and you can't take it away. So I think these are 2 very striking things about what you've heard today. I'm going to open up the mic for questions. Please, before you ask your question, state your affiliation and make sure you speak into the mic so that the reporters online can hear you. Questions? Yes.
Hi, Sherry Boschert with Clinical Endocrinology News. Dr. Spencer-Segal, could you comment, sort of taking all these as a whole and Endocrine Society policy or any public policy out there, what can physicians tell their patients to do or not do to reduce their risk? So much of this is prenatal exposure. I mean, do we need to get rid of carbonless receipts? Do we need to get rid of those linings and cans? What can doctors say to patients?
That's a very interesting question, and I also want to open it up to our speakers today. I'm going to say that I'm not prepared to make a policy statement today, but I do know that there are ways to reduce your exposure to these compounds, and especially for products for babies. I know that there are a lot of bisphenol A free products out there, and things are being labeled, so I think the awareness is there and that it's possible to reduce your exposure.
Gail Prince, the University of Illinois at Chicago. I would just like to chime in and agree with that statement. There have been some controlled studies that have looked to see when people use BPA-free products and go on a diet and a lifestyle for a very controlled period of time, they have been able to decrease their levels of BPA, but not completely. And so they're in products that we're not completely aware of, and we've not been able to eliminate it. And so it needs to be a two-pronged approach, one of taking steps to reduce your daily exposure if you know where it's coming from. And we also need more regulation across the globe in terms of reducing the use of this chemical in products and in consumer products. Let's just do it broadly, and to reduce the usage of this chemical so that we can get our levels down to where they are eliminated and they don't reprogram disease.
Yes, Patrick Fénichel. I am an endocrinologist, and I'm also gynecologist, and very often the women ask for the same question, what they have to do. I think a very important period, as everybody has shown here today, is during the pregnancy. And I think the physician has to say to the pregnant woman some recommendation. For example, you have not to use plastic containers in the microwaves, because you have a release of bisphenol A with high temperature. Something not to use plastic with bisphenol A. It's quite difficult because you have bisphenol A and plastic everywhere. But maybe during pregnancy you have to be particularly attentive. You know, in France we have now, the parliament forbidden the baby bottle, plastic baby bottle, and in 2 years we will try to not to use bisphenol A, but it's very difficult in practice.
Yes.
Other questions?
Sarah Allen from the Lancet Diabetes Endocrinology. Dr. Fénichel, I had a question about your study. You noticed that you had 52 boys born in the you had 180 boys born with undescended testicles, which is higher than the average that you quoted of 2 to 6%. Why is that?
No, it's not. We had 100 during this study, 105 cryptorchidism cases. It was in our array low percentage. We had at birth 1.8%, and after 6 months 0.8%. which is one of the lowest rate reported in the different countries.
Okay, I just have one more question. That's that because testosterone was not affected, does this suggest that it's the first phase that you talked about that is affected by BPA, potentially?
Yes, it's a good question, but it's more complicated because it has been demonstrated recently in rodents that INSL3 also act in the second phase. So we believe that our decrease of INSL3 is also a reflect of what happens at the second phase.
Thanks.
Next question.
I'm Gregory Goodwin from Children's Hospital in Boston. I understand the BPA is being replaced, so if it's BPA-free, they're replacing them with a substance that has similar bioactivity to BPA. Is that true?
Gail Prince, and so we don't always know what the industry is using and putting in products. And so I do know that there are substitutes that are being used now. Some of them are bisphenol compounds, and so bisphenol F, bisphenol Z, there are bisphenol S, and so there are a number of compounds that are those compounds are being substituted, but we don't always know what industry is introducing. These other compounds have not been thoroughly evaluated, certainly to the extent of BPA, but studies are coming out that they could disrupt other signaling systems, including glucocorticoid receptor pathways. And so this behooves the government to push through stronger regulations such as TSCA reform, whereby chemicals that are introduced into industrial and commercial use be first screened for their safety. And we need to push for that type of legislation before we become the guinea pigs to determine if they're safe, if we can indeed have a system in place worldwide and in the United States where we screen the compounds that we're replacing the bad players with. We don't want to go from bad to worse.
We have a question from the webcast from Ken Boyd from Access Medical. This is for Ms. Kennedy. Are there other products containing triclocarbons beyond personal care products? And based on your study, do you recommend these products be avoided? He also asked if you've conducted any studies on triclosan.
We haven't worked with triclosan. I know that triclocarban is in hospital sanitizers as well. There's a time and a place for antimicrobials. So when you're at high risk of infection, places like the hospital setting, daycares, nursing homes, things like that, it's important. However, in the general setting, we know that hand washing with soap and water is just as effective as using an antimicrobial soap. And so I would say that there's a time and a place for it, but it's important to know what the risk is and when it's needed.
Any other questions? No? Okay. I guess we will wrap up this news conference. Thank you all for attending, and I think our speakers are available if you have any other comments or questions for them. Thank you.